Hi everyone. In this module, I'll be discussing about enzymes. Uh, the objective of this module is to study the definitional properties of the enzymes, to study about the source, preparation, uses of enzymes obtained from plant source, animal source, and microorganism. Enzymes is a biocatalyst which catalyzes all the biochemical reactions taking place in all the living organism, and it is obtained from all the living organism like plants, animals, and microorganism. And all the enzymes are protein in nature and highly specific in actions, and it's sensitive to heat and pH. And the enzymes, based on the type of reaction it catalyzes, it can be classified as the enzyme which catalyzes hydrolytic reaction is known as hydrolases. Enzyme which catalyzes the transfer of chemical group from one molecule to another is known as transferases. The enzyme which catalyzes oxidation reduction reaction is known as oxidoreductases. Enzymes which is responsible for intermolecular arrangements is known as isomerases. Enzymes which catalyze the addition of groups to a double bonds is known as lyases, and the enzyme which catalyzes the condensation of two molecules is known as synthetases. Apart from this, based on the substrate on which they act, it can be classified as the enzymes acts on carbohydrate is known as amylases, example diastase, maltase, or invertase. Enzymes acting on the proteins is known as proteolytic enzyme, example pepsin, pancreatin, and papain. Enzymes acting on lipids is known as lipases, and the enzymes acting on nucleic acid is known as deoxyribonucleases. The plant enzymes which can be studied is papain. It is obtained from the latex of the unripe fruit of the plant Carica papaya belonging to the family Caricaceae. From the matured unripe fruit portion, a vertical incision is made at the interval of 1 to 7 days. The white latex which comes out is collected in a aluminium trays, and immediately after collections it should be treated with antioxidant like potassium metabisulfate. It should not be exposed too much to the sun because it gets decolorized. So after collections, it has to be the extraneous matter present in the lattice has to be removed, passed through the sieve, and the lattice is dried in a vacuum at a temperature of about 55 to 60 degrees centigrade. And papain is obtained as a white colored or light brown colored amorphous powder with characteristic odor and taste. It's a proteolytic enzyme which is protein in nature, and it consists of 212 amino acids stabilized by 3 disulfide bridges. And it is soluble in water and glycerin, and it is having the optimum activity at a pH of about 5 to 6. And the latex of this papaya fruit contains a mixture of enzymes like papain, chymopapain, caricane, glycyl endopeptide, and it is both a proteolytic enzymes and also amylolytic enzymes, so it can digest both carbohydrates and proteins. Because of that property, it is used for the treatment of digestive disorders, and it is also used to relieve the symptoms of episiotomy. It's a condition in which a small surgical incision is made during delivery in the vulva region. Also, it is an anti-inflammatory agent. Apart from the pharmaceutical applications, it is used in textile industry for degumming of silk fibers, in leather industry for dehairing of animal skins and hides, and in the beverage industry for clarification purpose, and in the manufacture of cheese as a substitute for rennet. And peptide is identified by adding a small amount of peptide to the milk. It becomes curd. The effect is known as curdling effect. And the second test with the potassium permanganate, it get decolorized, which indicates the presence of peptide. The next enzyme which is obtained from the plant source is bromelain. It is obtained from the juice of the pineapple fruit, Ananas commosis family, Bromelaceae, and both from the fruits and stem portions the bromelain can be obtained, and the enzymes which it is obtained from fruit, it is known as fruit bromelain. If it is obtained from stem juice, it is known as stem bromelain, and usually it is obtained from the pineapple juice, and it is treated with either acetone or with ammonium sulfide, which precipitates the enzyme bromelain. And it is bromelain is insoluble, incomplete is soluble in water, and bromelain enzyme, if it is obtained from fruit, it is acidic in nature, and the bromine derived from stem is basic in nature, and it is a glycoprotein with a molecular weight of about 25,000 to 31,000. It is mostly used to dissolve the clot, that is fibrin, in the condition of inflammatory edema. It is also used for meat tenderization, and baiting reagent for hides, which means the soaking of hides in the alkaline broth, and it is also used for production of protein hydrolase. The next enzyme is streptokinase. It's a bacterial protein obtained from the C group of strains beta amyloidic Streptococcus griseus. It's a the enzyme obtained from the microorganism. Actually, yeah, it is produced in the form of a lyophilized powder, and it is produced by fermentation process used by using the streptococcal culture, and it is bacterial protein with a half life of 23 minutes, and mostly it is a plasminogen activator. It has nearly 484 amino acid residues. It is used for dissolving the blood clot. So it is a plasminogen activator which converts the plasminogen to plasmin. This plasmin in turn dissolves the blood clot called fibrin, and it make into a fragmented degradation products called fibrin. So, because of that property, it is used in the treatment of pulmonary embolism, the condition which the blood clot in the artery of the lungs, and it is also used to treat venous and arterial thrombosis and for the treatment of coronary artery thrombosis. The next enzyme is urokinase. It's a serine protein enzyme obtained from the human urine and kidney cells, and by tissue culture technology and also by recombinant DNA technology. By using recombinant DNA technology, it is obtained from the genetically manipulated E. coli cells, and it is exists in two forms, that is a single peptide and another one is a double strand peptide. The first produced is a pro-urokinase, which is a single stranded peptide nucleus, and it is after isolation it is added to the adsorbents like silica gel or kaolin, which can be a purified product, which can be used as a purified product. It can also be further purified by precipitation with sodium chloride or alcohol, or even chromatography technique is also used for purification purpose. So, as it is supplied from the human source, it needs the sterile filtration and aseptic filling. It should be a sterile filling and also freeze drying products. As I said, urokinase occurs in two forms. One is a single polypeptide chain form, another one is double polypeptide chain form. Single polypeptide chain form has it's a low molecular weight, and double peptide chain is a high molecular weight polypeptide forms. And a single chain is produced by recombinant DNA technique, and it is also known as SCOPA, the single chain urokinase plasminogen activator. And it is similar to the streptokinase. It is also used to dissolve the blood clot. It is used in treatment of pulmonary embolism, coronary artery thrombosis, and for restoring the potency of intravenous catheter. The next enzyme is ceratiopeptidase. It's a proteolytic enzyme isolated from the bacteria Serratia E15. It can also be obtained from the silkworm in the larva stage. Ceratopeptidase is produced by fermentation technology using the enterobacterial strain Serratia E15. And during the life cycle of silk, the silk moth in the larva stage produces the enzyme in the intestine which break down the cocoon walls. So at this stage, we can also be obtain this enzyme from the silk moth larva stage. And selectivepeptidase is highly vulnerable to the degradation in the acidic pH. So when it is given in the oral form, it has to be protected from the acid in the stomach. So it is available as a entry coated tablet, which facilitates absorption to the intestine. It's the proteolytic enzyme which consists of 7.1 units per mg per solid. It's mostly prescribed as an anti-inflammatory agent, mainly used to treat the edema and swelling, inflammation, edema and swelling. Also, it is used to liquefy the pus and also sputum. It is used as a wound healing agent, fast wound healing agent. It can also be used as a alternative to NSA, non-steroidal anti-inflammatory drugs, and can also be used to treat rheumatoid arthritis and osteoarthritis. Apart from this, it is you having a wide applications in surgery, trauma surgery, plastic surgery, and respiratory medicine and gynecology. The next enzyme is pepsin, which is obtained from the stomach lining of the animals like pig, sheep, or calf. Commonly, it is obtained from the species of Pixascrofa belonging to the family Zoidae. Here, it is obtained from the mucous membrane, which is separated from the stomach by stripping of a scrap method, and it is placed in the acidified water for autolysis for 2 hours. After the two autolysis is completed, the liquid contains the enzyme peptone and pepsin. It is filtered and it is treated with either sodium or ammonium salt uh, until it becomes an half saturation. At this point only, the enzyme pepsin will separate out away from the peptone. Peptone will remain in the solution. The precipitated pepsin is collected and subjected to dialysis for the separation of the ammonium and sodium salt. If even some amount of pepsin will, if it is remain in the aqueous solution, can be pre-isolated by precipitation with alcohol. After the collection, it is dried and stored at a low temperature. It occurs as a pale yellow color, odorless, with faint odor and slightly bitter taste. It is soluble in dilute acid, and it is having a best activity at a temperature of about 40 degree with, and with the pH optimum pH of action is 2 to 4, and it get denatured above the temperature of about 70 degree. Also, alcohol and sodium chloride will denature the pepsin, and it can be stored for 2 years at even for two up to 2 years at 2 to 8 degrees centigrade. Mainly, it is used in the treatment of deficiency of gastric secretion, and it is also used in the analysis of proteins and the preparation of cheese. Thank you for watching.